Hello, my name is Taylor Fraser, and welcome to my presentation on apoptosis and hibernating Tenrec ecaudatus. Before I begin, I just wanted to say thank you to the National Institute of General Medical Sciences for the grant that funded this project, and another thank you to the Tenrecs, Dr. Van Brooklyn, Michael Treat, and Gilbecca Smith for making this research possible. Tenrec ecaudatus, or the common tenrec, is a small placental mammal that is native to Madagascar. These nocturnal Afrotherians exhibit several ancestral traits, such as a small and smooth brain, cloaca, and variable body temperatures. During the astral winter, tenrecs typically spend 8 to 9 months in hibernation, in which they are consistently torpid, but the state of torpor can vary. Some tenrecs are more lethargic or more active than others. Torpor is a state that animals experience where oxygen consumption is reduced to below what we typically think is needed to maintain homeostasis. Ground squirrels are an excellent example of a classic hibernator. Ground squirrels will experience torpor bouts of roughly 8 days, then undergo an interbout arousal. Interbout arousals are a period of euthermic activity where organ functions, cellular processes, and metabolisms are resumed. These periods typically last for 12 to 24 hours and potentially help animals recover from the consequences of torpor. Tenrecs lack interbout arousals. So the purpose of this project is to understand how apoptosis is processed during hibernation in tenrecs. Apoptosis is an energy-demanding cellular mechanism to events like oxidative stress, hypoxia, and acidosis that occur during hibernation. The invocation of widespread apoptosis or programmed cell death would be detrimental to an animal that is conserving energy like a hibernator. The degree of apoptosis compared between active and hibernating tenrecs could explain the idea of how much cellular stress is being sustained during torpor, and how that damage is potentially processed in atypical hibernators. Analysis of apoptotic rates with contrasting metabolic activities or designated temperatures could speak to tenrec's lack of homeothermy. 12 livers and 12 kidneys were collected from active and hibernating tenrecs housed at 12 and 28 degrees Celsius. Specifically, 3 from each group were collected, so for example, 3 livers were collected from tenrecs housed at 28 degrees Celsius who had a body temperature of 28 degrees Celsius and were in an active state. Another group that was collected are tenrecs that were housed at 12 degrees Celsius who had a body temperature of 12 degrees Celsius and were in a hibernating state. These organs were prepared for staining by sectioning the tissue at 1 cell layer thick, or roughly 6 microns, using a cryostat. Then a terminal deoxynucleotide transferase TDT-DUTP nick and labeling assay, also known as a tunnel assay, was performed on 3 prepared tissue samples per organ. This determines if the number of apoptotic cells could be associated with metabolic activity or temperature. A tunnel assay is able to attach a fluorescent marker to Nixon DNA, determining if apoptosis had occurred or not. Fluorescence microscopy will be used to visualize and quantify non-apoptotic and apoptotic cells. The tunnel pictures presented are actually all ground squirrel tissues. Pictures A and B are tunnel-positive kidney cells. Picture C is a positive control for the kidney tunnel assay. Pictures D and E are tunnel-positive liver cells, and picture F is a positive control for the liver tunnel assay. The remarkable thing is that on average, a hibernating ground squirrel will have between 0 and 1 apoptotic cell in a given field. During interbout arousal, that number only goes from 2 to 5. Recent work in the laboratory details how ground squirrels actually mitigate apoptosis during hibernation. However, tenrecs lack interbout arousals, so I hypothesize that tenrecs will experience much higher levels of apoptosis when colder or during hibernation. This concludes my presentation, and I just wanted to say thank you again to the National Institute of General Medical Sciences for making this project possible. This has been an incredible opportunity, and I have grown immensely in the past 10 weeks. Thank you all for listening.